Log on, tune in, find out. Another good idea from Cambridge.
This morning's session is Daniel Doerr from EPFL University. He's talking about stochastic errors versus modeling errors in distance-based phylogenetic reconstructions.
So thanks for the introduction, Daniel. My talk is about joint work with Ilan Kronau from Cornell University and Shlomo Moran and Irat Yavne from the Technion. And in our project, we studied distance measures. So I quickly want to give you a brief overview about the whole reconstruction process. I guess you are familiar with it anyway, so I make it really short. Starting off with a model tree, you assume that sequences evolve along the edges according to some substitution model. We can only observe the sequences at the leaves. From those sequences, we can construct an alignment, and then the actual reconstruction process starts. We first choose a substitution model, which we assume is the underlying one that led to the observed substitutions. And then we choose a distance measure according to our chosen substitution model, and then we calculate all pairwise distance estimates using our distance measure and feed this into a tree reconstruction algorithm such as neighbor joining or whatever your choice might be, and we will eventually get a tree. And we hope that this tree will be an unrooted representative of the model tree along which the sequences evolved. So in this project, we are interested in distance measures, and we wanted to find new ways in helping the user to make the right choice in choosing the distance function, because the performance of those distance functions vary greatly, and this results in different results. This results in different accuracy in reconstruction. So this work is a follow-up on different efforts in this area from Ilan, Shlomo, and Irat. But this time we didn't only look at consistent distance measures, but also considered potentially inconsistent distance measures that, and we wanted to predict cases in which potentially inconsistent distance measures actually improve accuracy compared to consistent ones. So just a quick introduction. I spare the details because of lack of time, I guess. What are distance measures? Simply and generally speaking, a distance measure is a function that estimates a distance between two sequences. And in phylogenetic reconstruction, we usually use additive distance measures because in case our distance estimates are accurate, this results into a tree metric, the tree metric of our model tree. And this is the connection between consistency and additivity. So when we are using additive distance measures, we will get the correct tree if the estimates are correct, and that's why it's called consistent. And for potentially inconsistent methods, those are not additive. We might find cases in which the model tree cannot be reconstructed. So in homogeneous models, distance function can be represented as a function of the evolutionary time t. And I want to show you some simulations, and in those simulations I use the Kimura 2-parameter model, which is a very simple model. It has only 2 parameters. It differentiates between transitions and transversions. I think we heard this before, so I'm not going into the details. Usually the rate parameters are normalized, so we can infer some uniform measure of evolutionary time. And for the K2P model, there exist many different additive distance measures, which are also called substitution rate functions in the context of substitution models. So if you see some SR here, this basically means substitution rate function and is synonym in my talk for distance measure. So I'm only using one additive distance function out of the many which I could choose from, and I use the standard distance measure, which simply estimates the evolutionary time. So here is a simple experiment. We use a model tree, which is called Hasegawa's tree. It has 7 taxa. And this tree is challenging to reconstruct because it consists of both long and short edges in the tree. So we discard the underlying genetic sequences that led to the reconstruction of this tree and generate new ones according to our K2P model. We use a transition to transversion ratio of 2. And we scale the tree into different sizes. For each size, we generate 10,000 batches of alignments. And for each batch, we try to reconstruct the tree. After we reconstructed the tree, we measure the Robinson folds distance to the model tree, and this is what is actually plotted here. So at the beginning, we see that only few substitutions occurring, so this leads to noisy distance estimates, and the reconstruction is bad. But with increasing tree diameter, there are more substitutions occurring, and eventually there are so many substitutions occurring that the sites saturate, and this leads to multiple substitutions, and the distance estimates become noisy again. We did this experiment also with a different distance function, which is the Jukes-Cantor distance, and we were surprised to see that the accuracy is actually increased, although the Jukes-Cantor distance assumes the wrong model. It assumes a model in which r equals 1/2, and here we use 2. So we wanted to investigate into the behavior of JC, and so you can look at the single distance estimates of both functions. We see that the standard function is pretty much linear because, well, it estimates the evolutionary time, but the JC or Jukes-Cantor distance is concave, and it kind of like looks that with increasing evolutionary time, the distance estimates become inaccurate. But if you go back to our original experiment, we actually see an increase in improvement compared to the standard method, especially for larger tree diameters. So we think that this behavior can be explained by 3 different factors. One is that if you look at the variance of the 2 distance functions, we see that the standard function has increasing variance in increasing evolutionary time, whereas the Jukes-Cantor distance has a very narrow variance. So this is the first factor. The second is that in phylogenetic tree reconstruction, the distances we try to estimate are quite limited. They are limited by the largest and the shortest distance in the tree. So a distance function only needs to be additive in a quite narrow range. And the third factor is if we look at the linear interpolation of our non-additive distance function with the Jukes-Cantor distance in this interval, we actually see that it's a quite good approximation of our the distance in this interval. So the interesting observation here is now that this linear interpolation actually represents an additive distance function, because every affine transformation of a of an additive distance function is an additive distance function. So we could actually use the standard distance function, transform it onto this line, and this what we did. We tried to actually minimize the difference between our non-additive and additive distance function, and if you combine all those 3 factors together, you see that although the Jukes-Cantor distance is biased, that's I think so. This is actually the error. The red error here is actually the error that originates from the fact that this distance is potentially inconsistent. It assumes the wrong model. However, this disadvantage is basically overruled by the gain in accuracy, which results from the lower variance in this area. So we would actually choose the inconsistent or potentially inconsistent distance compared to the consistent one, because it has a general lower estimation error in this interval. So this led to a concept which we call a deviation from additivity. We can measure the deviation of our inconsistent distance function in the interval of reconstruction, and we can use this measure for two different things. First, we can check for consistency, so we make sure that our model tree can actually be reconstructed using our non-additive distance function, and then we can also use it to choose the candidate with the overall minimal estimation error, and the estimation error does not only consist of the deviation from additivity, but also from the variance. So can we actually predict the performance of non-additive distance functions in tree reconstruction? And this is what we tried to do with quartet trees. We constructed a framework which allows to exactly do this. Here are two quartets with long and short edges. I think you see those before in previous talks. So that's the Felsenstein quartet related to the Felsenstein stone and the Paris quartet. And the long edges in this tree are 5 times larger than the short edges. So what happens is that the non-additive distance, the Jukes-Cantor distance, which we used in an experiment here, underestimates the sums of the long and the short distance in the tree. So if we compare it to our additive distance, we see that our additive distance pretty much behaves equally well in both cases. However, the performance of our non-additive distance function is quite different. So here it is actually worse than the standard distance, and here it basically gets the tree always right. So, but even in this case, in the Felsenstein quartet, if you use moderate instances of the Felsenstein quartet where the large edges are not 5 times but 3.5 times larger than the short edge, we see that it actually gives a better result than the standard distance. So we use the Fisher's linear discriminant in order to predict the performance of non-additive distance function in homogeneous models. The Fisher's linear discriminant measures the separation between independent normally distributed random variables. We don't have normally distributed random variables, neither are they independent, but it gives a quite accurate estimation of what actually happens in quartets, and that's what I'll show you in an experiment on the next slide. So using the Fisher's linear discriminant, we try to estimate the ability of non-additive distance functions to separate along the true split of the quartet, and this is what we simulated in this experiment on 112 trees per data point and with a sequence length of 1,000 base pairs. We used the Felsenstein quartet and gradually decreased the size of the external small edges. So this is what you see here, and we see that at some point the performance of the additive distance function here is better than the non-additive one, the Jukes-Cantor. And we can actually predict the same behavior using our FLD method, and also this equilibrium point can be predicted fairly well with FLD. But there's another potential or in using this method, and this is and we can use actually this method to compare different distance functions using two types of quantities that we get out of the FLD. One is the separation, and the other is the noise. And if we perform this, comparisons between different distance measures, this performance is actually independent of the sequence length. So this is another experiment with the same Felsenstein quartet with gradually decreasing short edges, and we see that the noise ratio between our non-additive distance function and the additive one, so JC versus K2P, is remaining rather constant, whereas the JC's ability to separate along the true split decreases with decreasing shorter edges. So we can predict this behavior pretty well in simulations, but we also performed a biological experiment which we think supports our case. So we used 163 bacteria species and extracted 31 marker genes out of each single one of them. Then we sampled 40,000 sub-alignments and extracted the fourfold degenerate sites, which we then used for distance estimation. And we compared the reconstructed tree against the reference tree, which we obtained from the Living Tree project of Arp Silva. And this tree has been constructed using 16S RNA. So we compared three methods against one which we think is probably the most additive one in this alignment, and for this we used GTR plus invariant sites and rate variability, so gamma distributed rates across size, and we reconstructed the tree with this distance measure using PhyML, and binned the reconstructed tree. So we compared it against the reference tree using the Robinson false distance, and then we binned the trees according to their Robinson false distance along the x-axis here. And then we reconstructed for each bin the trees again using our three methods which we wanted to compare. And we see that using a very oversimplified model here, Stokes-Cantor, we actually obtain better results than using log-dead, which is very close to additivity in this kind of setting too. So, our message is basically that using distance measures that originate from oversimplified models, you can actually gain accuracy if if they still remain consistent in our model tree. So to summarize, we had this surprising observation that non-additive distance functions actually can gain accuracy in compared to additive distance functions. And yeah, we did experiments on K2P trees. We introduced a framework which allows to analyze non-additive distance functions, and more information is available in our Wabi paper that has been submitted. So
We have time for a question.
What sequence lengths did you use, and will the result change for longer sequences?
In the simulations or in the
simulation? In general.
yeah. Well, it's actually the performance is independent of sequence length. I mean, the non-additive distance function will always be better than the consistent ones. Potentially, yeah.
I also had a question.
Yeah.
Instead of using Jig's hand-away, just use Hamming distances. I mean, that's
Yeah,
ridiculous.
we could have.
Did you look at that at all?
No, we didn't, but yeah.
Okay, well, thank you once again.